All right. Assalamu alaikum. I hope all of you are fine, my dear sweet students. Today we will begin with kidney. So it is a chapter of kidney. We will begin today with me, Dr. Nadia Salam, and we will deal with topics one by one. Uh, the most important and difficult topic in the entire chapter is glomerulonephritis because immune deposits in particular deposit in one or the other layer of GBM, glomerular membrane, basement membrane. GBM is glomerular basement membrane, and they trigger a different sort of glomerulonephritis, each having its own characters, like they may or may not respond to the treatment, right? So, what we are dealing with, we have to diagnose what kind of glomerulonephritis we're dealing with, and what is all this glomerulo. You know what is glomerulus? Oh, you know, what is all this glomerulonephritis? Now, the glomerulus is the tuft of capillaries present in the Bowman's capsule. You know we have two kidneys, and you know anatomically they are present in the rib cage or behind the rib cage in the abdomen, in the abdominal area, right? In the upper abdomen. And these two kidneys have got functioning or functioning units. These functioning units are called as nephrons. So study of a single nephron gives us idea of the whole kidney function. So nephron is a kidney unit. Once destroyed, does not proliferate an entire nephron by itself. So loss of a single nephron means that you have lost a kidney unit, right? Do we need two kidneys? Actually, yes, we do need two kidneys. Anything can go wrong in one kidney, the other kidney makes up for the deficiency. So you don't keep throwing away your kidney, right? Don't just keep it, keep both the kidneys. All right. So that is how nature works. Whatever He's given us is good for us. Nothing is extraordinary. Nothing is that extra to uh, to spare one. All right. So we will talk in detail why I'm saying that in scientifically. So I was saying that a single loss of a single nephron means that a single kidney unit has been lost. Glomerulus diseases of glomerulus are blood vascular diseases that affect the kidney tuft of kidney that is inside the Bowman's capsule. So, what can disturb this glomerulus? Either it could be autoimmune, or it could be infection, or it could be another trauma, or there could be something very much focal, specific for glomerulus alone, right? Some immunity or something that hits the glomerulus alone. Why is it? Why is it so? Because the entire blood comes to get filtered in the glomerulus, so it is prone to get some kinds of injuries, and it has some features which make it more prone to get injuries. So, yes, Intizamul Haq. If there are any questions by your students, please do write to me, so I will try to clarify all of them. So, yes, we were talking about if there's an injury to the glomerulus itself, because structure is such, and or it could be some generalized disorder of the vascular system of the entire body. So, if some disease that affects the entire vessels of the body will affect the glomerulus, definitely it will affect the glomerulus because it is capillaries after all, it is part of the vascular system. So one of them, for example, is hypertension. The other, for example, is diabetes mellitus that will affect kidneys or glomerulus or blood vessels of the kidneys, right? So let us see the structure and let us see what happens to the kidneys when it has been struck or when it has been destroyed or whatever goes wrong with it and what can go wrong with it. So there is something very special about glomerulus, right? This network of capillaries have got something very special. What is that special? It is fenestrated, which means the endothelial cells, the cells have got pores in them. No cell in the body has got pores in them. And these cells are fenestrated, they have got pores so that things can flow out or sieve out of them, right? Anastomosing network of capillaries have got uh, some epithelial cells attached to them, and those epithelial cells belong to the Bowman's capsule, and they are divided into visceral and parietal epithelial cells, right? So here is the entire Bowman's capsule. This is the parietal layer of the Bowman's capsule. This is the Bowman's space. And this is the visceral layer of the Bowman's capsule. So the visceral layer of the Bowman's capsule again has got a special structure that causes food processes to protrude out. Those food processes are called as podocytes, or podocytes are called as the second name of the visceral epithelial layer is also called as podocytes, or the food processes are protrusions of the podocytes, right? And then there are proximal tubules, and then there are distal tubules. So we will study problems which each with each of the structure of this nephron. So first we will begin with the Bowman's capsule, and then we will keep going on to the tubules and then see what goes wrong in the tubules and how you will differentiate problems of the tubules from that of the Bowman's capsule and from that of the glomerulus. So it has got a fenestrated endothelium, porous endothelium, an anastomosing network of capillaries, and then it has got visceral epithelial layer. Visceral epithelial cells are same as the podocytes. The cells are called as podocytes, visceral epithelial layer. They are incorporated into and become part of the capillary wall. Yes, definitely we have seen that. Look at those podocytes. They are incorporated and they become part of the endothelial wall. And then we say they are separated from endothelial cells by GBM, glomerular basement membrane. Glomerular, this thick dark pink color is the glomerulus basement membrane. Glomerular basement membrane has got tremendous function in the kidney, or it has got a lot of significance in the kidney. In other places, we don't find that many diseases of the glomerular basement membrane as we find in the kidney. The parietal epithelial layer, situated on the Bowman's capsule, lines the urinary space. This is the parietal epithelial layer. You can see it is a simple epithelium that forms a cover of the glomerular tuft of capillaries. Afferent arterioles are the incoming arterioles that provide blood. Efferent arterioles are the outgoing arterioles that take away blood. So another difference. We said that the endothelial layer of the capillaries, the cells of the capillaries have got pores called fenestra. And now we're saying efferent arteriole supplies blood and efferent arteriole takes away blood, which means glomerular tuft is located between two arterioles, not between vein and an artery. So this is the second difference. Two arterioles are there. Two arterioles, one is meant for giving the blood, other is meant for taking the blood. So it means it is a very high pressure system. Veins have got low pressure and arterioles have got high pressure. So something, a tuft of capillaries, when it is located between two arterioles, means that it has got a very high pressure system. It is a very high pressure system. So this is the Bowman space where the filtered material comes in, and filtered fluid flows into the tubules, and the entire process forms the urine. In a normal condition, this is mesangium. This is mesangium. Inside the mesangium, you can see green colored cells. They are mesangial cells. Mesangial cells come from the mesenchyme, which is connective tissue, and mesenchymal cells are the connective tissue of the cells. They have got their own significance, right? So we will go through what is the significance of mesenchymal cells as well. Podocyte, right? And these are the foot processes of the podocyte. Look at the endothelial cell, endothelial cell, and this is the lumen of the capillary, endothelial cell of the capillary. It has got so many slits, it is broken in places. You know the size of the slits? Yes, I know you know. It is 70 nanometer, right? So there's something very strange. 70 nanometer is the size of the slit, and albumin passes through it. Can it? No, it cannot. And what is the size of albumin? Some books say nanometer, others say it is 3.8 nanometer or 4 nanometer, which is far too small than 70 nanometers. So why does it, or why can't it pass through the porous or fenestrated endothelium of the capillary? Why can't it pass? Because there is another barrier there. See? So another barrier that is not actually visible. There are negative charges here. There are negative charges here. Albumin is a protein and it has got negative charges as well. So they repel each other and it cannot pass out of the glomerular basement, or it cannot pass out of the endothelium. The only way it can pass out is once the charges are nullified, right? If the charges on the endothelial cells and the glomerular basement membrane are nullified, it will pass through this barrier. Please look at the interdigitating process of the podocytes or the foot processes. These are called as pedicles. Interdigitating processes of the pedicles. There is something very special I will explain, because there is some connection between these pedicles as well. This is the diagram in your book and how you will read it. This is the epithelium or the endothelium. Endothelium has got so many pores in it, right? This is the foot process, blue color foot processes, and those are the pedicles. In between them is a pink layer. You can see pink layer. The pink area is the basement membrane, the glomerular basement membrane. And this here is the parietal epithelial layer, right? So this is the space where the urine is supposed to form. These two cells are mesangial cells. They lie in a green area of connective tissue, or you can say a sort of semi-fluid area. This is mesangial matrix. This is the matrix area. And this together is glomerulus, right? If you understand what is normal, then we can talk about what is abnormal. If you don't understand normal, then understanding abnormal becomes very difficult. Some of you have got picture memory. They pass their exam wonderfully because they can mug their things well, but that is not the way things work in your patients. Unless you've got good understanding, things will not work out. So let us just create understanding. Glomerular capillary wall is filtering membrane because it is fenestrated, it has got pores, therefore it filters. And these pores are 70 to 100 nanometers in diameter, 70 to 100 nanometers in diameter. So which means you can see this is larger pores, you can see this is smaller pores, something in between 70 and 100, but the range is like that, 70 and 100 nanometer in diameter. This is an RBC, it is far larger, it cannot pass through the fenestra. Just look at the capillary, it has so many pores in that, small and large, 70 to 100 nanometer, right? So uh, this is again an RBC that cannot pass through the uh, endothelial cells, and look at the basement membrane that also encloses the that goes around the endothelial cells. This is electron micrograph. This is what podocytes or food processes look like, right? This is what podocytes look like. This is what endothelium looks like. Porous endothelium. It has got so many. Look at this basement membrane. The basement membrane differs from the rest of the basement membranes in the body in that it has got 3 layers, right? So in between, you can see a dense layer. This is lamina densa. Around it, you can see lighter area, lamina rara. This is lighter area, lamina rara. So the facing area facing in the capillary lumen is lamina rara interna. The one facing outside or the one facing the porocyte area is lamina rara externa. Lamina rara externa, lamina rara interna, and in between lies the dense layer of the glomerular capillary wall. So when we say GBM, glomerular capillary membrane, we say that it is a very large area. What do you mean by GBM? Where is the injury located? Is it on externa, interna, or is it in the dense layer? Right. So the injuries go like that. GBM, glomerular basement membrane, a thick electron dense central layer, the lamina densa, we have done that. The thinner electron lucent peripheral layers is lamina rara interna and lamina rara externa. So here is another diagram. This is interna. You're right. So this is interna, and this is lamina rara externa. Why? Because there are food processes here. See? So there are food processes here, and this is lamina rara externa. And there are endothelial cells here. You can see fenestra, and this is lamina rara interna. In between is a blue area, which is the dense layer of the lamina densa, dense layer of the GBM. Note another thing. Just note another thing in the diagram. Do you think a gray line attaching to podocytes? Do you see a gray line attaching to podocytes? Okay? This gray line attaching to podocytes. It has got an ultra structure as well, and this forms the diaphragm by itself. So when blood comes into the capillary, it passes through the filtration slits, or it passes through the fenestra. I'm sorry. So it passes through the fenestra, and after it passes through the fenestra and it crosses the charge layer, or it crosses the negative charges, it enters the GBM. It has to pass through GBM, and then it has to cross these. It has to cross these diaphragms, and it has to pass through the slits. These are called as slits. The space between two podocytes is slits. GBM consists of collagen, mostly type 4 collagen. So we have studied where type 1 collagen is located in the bones, where type 2 collagen is located in the cartilage, 3 is spread widely, 4 is under the floor, and under the floor means what is under the floor is GBM in the glomerular basement membrane. Laminin. Polyanionic proteoglycans, mostly heparan sulfate. Proteoglycans, you have studied that in biochemistry. Fibronectin and nectin, and several other glycoproteins. So these are all names of the glycoproteins. Type 4 collagen forms a network of superstructure to which other glycoproteins attach, right? Visceral epithelial cells. Well, I'm sure you know things a lot more than before about visceral epithelial cells. Podocytes possess interdigitating process. Yes, you know that. Podocytes are embedded in and adherent to the laminar vera externa of GBM. Yes, you know that. And we did not talk about the food process are separated by 20 to 30 nanometers wide filtration slits. These are bridged by a thin diaphragm. A thin diaphragm bridges these two podocytes. I think we can go back again. This is the bridge, right? A thin bridge. There is a thin bridge between two podocytes, and the space between the two podocytes is the filtration slits, which is 20 to 30 nanometers. Right, so 20 to 30 nanometers. These are bridged by a thin diaphragm. These are the endothelial cells, right? So the podocytes you can see here, right? Mesangial cells, yes, those cells are in the mesangium supporting entire glomerular tuft. They lie between the capillaries. Mesangial matrix is the basement membrane-like and forms meshwork in which mesangial cells are embedded. So we have seen this diagram before as well. There are three mesangial cells by the book. Three mesangial cells, two mesangial cells, or even one mesangial cell is normal. Anything above three mesangial cells could be inflammation. And what else can come in inflammation? Inflammatory cells, neutrophils, lymphocytes, and so many others. Mesangial cells are of mesenchymal origin, which means they're just connective tissue cells. They are contractile, phagocytic, capable of proliferation, lay down both matrix and collagen, secrete several mediators. Biologically, they resemble vascular smooth muscle cells and pedicles. They resemble smooth muscle cells and pedicles, but they are not smooth muscle cells. Mesangial cells are not smooth muscle cells because they phagocytose and they lay down matrix and collagen, so they're not smooth muscle cells. But they do resemble, there is a resemblance of mesangial cells with the muscle cells. They're important in many forms of glomerulonephritis. So please note here that these special cells of the mesangium that are connective tissue cells, they are acting as a phagocytic cell or an inflammatory cell by itself, which means it will protect against any pathogen or any foreign body that it can engulf. And it acts as a smooth muscle cells as well because it has got contractile power and it has got muscle cells, it has got pericytes. Let us examine in detail. This is endothelial cell, right? This is the Bowman's capsule, parietal layer, all right. This is a podocyte, the nucleus of a podocyte. Look at it, how it has attached itself to the endothelial cell. Then it is intraglomerular mesangial cells. Look at the two cells that sit in between, right? Intraglomerular mesangial cells. Those are the intraglomerular mesangial cells. Because of fenestrations in the endothelium, the normal glomerulus is permeable to water and small solutes. Yes, water doesn't have charge, so impermeable to proteins, even the size of this albumin, which your book says is 3.6 nanometer in radius. And or it can be slightly larger, but it is 3.6 nanometer in radius. The size and charge dependent barrier, right, prevents albumin from filtration as albumin is an anionic molecule, so there is a negative repulsion of albumin, so it will not flow out. Visceral epithelial cells, they maintain glomerular barrier function. Its slit diaphragm is a size-selective distal diffusion barrier to filtration of proteins, and is largely it is largely synthesizes components of glomerular basement membrane. It maintains filtration glomerular barrier function. Does it? Yes. Does it form any barrier function? It has got diaphragms, it has got slits, so the slits will act as a barrier function, or it will allow only certain size of the molecules to pass. And the diaphragm acts as a barrier, right? So it has got two barriers. So it has got size selection, right? 20 to 30 nanometers size selection, because that is the distance between the two podocytes. Here again, these are the podocytes, and these are the slit processes. These are the slits, and this is glomerular basement membrane, and these are the slits of the podocytes. So this is the amount of fluid that can pass through here. A whole red blood cell is going through diapetesis, right? A red blood cell is passing through diapetesis. This is capillary lumen. So this is the slit, and this is from where the fluid can pass out. But even in the slit, there is a diaphragm. The proteins located in the slit diaphragm or present in the assembly of molecules within the visceral epithelial cells are attached to slit diaphragm. So this is the ultrastructure of the slit diaphragm, or you can say what happens in the diaphragm of the podocyte. The barrier or the diaphragm, this is something proteins located here are called as the diaphragm, or you can say they form a diaphragm. And the ultrastructure of the diaphragm is significant because in case of its destruction, unwanted material or things we have to keep in the body flow out of the glomerulus, and they are lost in the urine. Present in assembly of molecules within the visceral epithelial cells that are attached to the slit diaphragm. CD2AP, right? CD2AP is a protein, right? Is then present in the molecules within the visceral epithelial cells, and CD2 associated protein. This is a podocyte associated CD2. So this is nephrin. A nephrin molecule forms the diaphragm, and look at the nephrin when it enters the podocyte. It is attached to podocin here. It is attached to actin here, and it is attached to CD2AP, CD2 associated protein, right? CD2 associated protein, podocin, actin filaments. This is what nephrin is attached to inside inside a pedicle, right? Inside a pedicle, and this is the ultrastructure of the visceral epithelial cells or a podocyte. Nephrin is a transmembrane protein. It has a large extracellular portion compared to a small intracellular portion, made up of immunoglobulin-like domains. Extracellular portion is immunoglobulin-like domains, right? This is immunoglobulin-like domains. And a nephrin molecule extends towards each other from neighboring food processes. Nephrin molecules extend from each other, right? Nephrin molecules extend towards each other from neighboring food processes. They dimerize across the slit diaphragm. So this is, they are passing through the basement membrane, right? This is the slit membrane, and they are dimerizing across the slit. They dimerize the slit diaphragm, right? And they've got a small intracellular area which is attached again to three other molecules. Within the cytoplasm of the food processes, nephrin forms molecular connection with porosine CD2 associated protein acting like cytoskeleton of the visceral epithelial cells. We have talked about that. Actin filaments, we said it is attached to actin filaments, porosine, and CD2 associated protein. So this is exactly what is meant here: porosine, CD2 associated protein, and actin like cytoskeleton of visceral epithelial cells. Most lentiform proteins are identified. Mutation in the gene encoding them give rise to defects in permeability and nephrotic syndrome. We will study nephrotic syndrome in a moment, and I'll just give you the definition of what nephrotic syndrome is and try to keep it in your mind exactly what nephrotic syndrome is. Glomerulopathies are characterized by one or more of the four basic tissue reactions. So whenever there is tissue injury, it is on microscopy. Uh, it is accompanied by hypercellularity, basement membrane thickening, hyalinosis, and sclerosis. Whenever there is an injury in the body, there is hypercellularity of the area, or the cell proliferates. The cell in the area proliferate to compensate for the injury, and then there is a lot of vascular leakage and edema formation, and then there is a lot of inflammatory cells that pour in to counteract the cause of the injury or the foreign body that has caused the injury, or bacteria or whatever. But what happens in the kidney? Hypercellularity, yes, it is just the same as the rest of the body. Basement membrane gets thickened, right? Now, if the basement membrane of the kidney gets thick, it has got very strong effects on the filtration. And hyalinosis and sclerosis. Hyalinosis means some amorphous material deposits in there. Sclerosis is narrowing of the walls of the of the capillaries. Sclerosis is narrowing. Look at this. This is normal. I said even one mesangial cell is normal, and I told you that this is the mesangial matrix where it lies, and this is what normal looks like, right? Endothelial cells and everything. Look at the mesangium here. 1, 2, 3, 4, 5, 6. There's supposed to be 3 at the most. So this is inflammation. Look at the basement membrane. Yes, nearly of the same size, but there is something wrong here. Increased number of mesangial cells, subepithelial humps. Look at this podocyte, right? Podocyte here, all clean and nice with its pedicles surrounding the basement membrane of the glomerulus. But something went wrong here. This is these are the immune deposits that have formed humps. So if immune deposit forms humps, that means there is a specific glomerulonephritis, right? So this is how we recognize glomerulonephritis according to the location of these immunological deposits, whether they are inside the glomerular basement membrane, whether they are in the subepithelial layer, or whether they are in the um, in the visceral epithelial layer, right? Subepithelial, subendothelial, or in the basement membrane, inside the basement membrane, and what they are forming, right? What they form. This is how we diagnose. Hypercellularity results from one or more of the following: how proliferation of the mesangial or endothelial cells, infiltration of leukocytes, formation of crescents. So we said proliferation of the mesangial cells causes hypercellularity, and I told you that there could be infiltration of the inflammatory cells as well among the mesangial cells that will cause hypercellularity. Then it says that there is proliferation of the endothelial cells as well. So endothelial cells could proliferate. These are the endothelial cells. These can proliferate and cause hypercellularity as well, which is not present in this diagram. But these cells also can be a cause of hypercellularity. Infiltration of leukocytes include neutrophils, monocytes, and occasional lymphocytes. Yes, those are the inflammatory cells. Following immune inflammatory injury, yes, how the crescents are formed. Yes, how the crescents are formed. I think, all right. Following immune or inflammatory injury following a capillary wall, the cells are composed of proliferating predominantly parietal epithelial cells, some visceral epithelial cells, and infiltrating leukocytes. Crescents is a moon, half moon is a crescent, and crescent forms in the glomerulus or in the in the Bowman's capsule, not actually in the glomerulus, because crescent is formed once inflammation is triggered. Let me show you. Once inflammation is triggered here, the Bowman's capsule, there is it, the Bowman's capsule, it also reacts. And how it reacts? It proliferates as well. It proliferates here. Inside the urinary space, it proliferates here. And when it proliferates here, it tightens the area or narrows the urinary outflow. And not only that, it obliterates the urinary space as well. So although all other cells are proliferating, but it is the proliferation of the mainly or basically it is proliferation of the parietal epithelial layer, right? That will induce a crescent formation. Just imagine that if the parietal epithelial layer proliferates, what will it cause? It will cause obliteration of this urinary space and decrease in the area of like. Just look at this one is your usual H&E stain and a usual microscope, right? So in this microscope, there is a lot of mesangium. There is a lot of mesangium. This is the Bowman's capsule, and in the Bowman's capsule, there is this urinary area, urinary space, and proliferation of the Bowman's epithelium has caused obliteration of the space, and it is obstructing on the glomerulus as well, right? So the area of urine outflow has decreased. The area of urine outflow has decreased. This is the Bowman space. Look at the whole parietal epithelium that has surrounded actually. This is the urinary tubule. This is urinary tubule because it has got all epithelial cells and it has got so many cilia inside. Right? So ciliated epithelial cells. This is proximal convoluted tubule, and this one is the glomerulus. The mesangial cells. Look at the mesangial cells. They have increased in number. Mesangial cells have increased in number, right? Plasma protein, formation of crystal plasma protein leak into the urinary space. They get exposed to procoagulants and tissue factors. This leads to fibrin deposition. Activation of coagulation factors such as thrombin triggers the crescent formation. Multiple pro-inflammatory cytokines recruit leukocytes into the crescents. So, what is going on? Cytokines are recruiting other leukocytes. There is proliferation of epithelial cells that causes crescent formations. Such things occur. This is a condition and not the name of a disease. Please keep in your mind, this is a condition, not the name of a disease. Plasma protein leak into the urinary space, get exposed to procoagulants, for example, tissue factors. This leads to fibrin deposition and activation of coagulation factors such as thrombin, which means activation of coagulation factors, which means there is coagulopathy in the body. It means thrombi will form in the kidney diseases, in the glomerular diseases. It is prone to through thrombi in case if there is proliferation, there is inflammation in this glomerular area. Multiple pro-inflammatory cytokines recruit leukocytes and crescents. So, basement membrane thickening. These changes appear as thickening of the capillary walls, best seen with, do you call it periodic acid shift? My all students have always read it as periodic acid shift. This is per iodic acid shift. It is an iodine. It is an iodine stain. All right, iodine stain. Therefore, it is per iodic acid shift. And why is it used? To detect glycoproteins. Since a lot of glycoproteins are leaked out of the glomerular capillary, out of the basement membrane. Therefore, it is called as per iodic acid shift. Electron microscopies, such as thickening, it takes one of the three forms. So we can use periodic acid shift to see all the proteins that have leaked out, or all the glycoproteins that have leaked out. Right. Marked global thickening of the glomerular basement membrane, which has vacuolated appearance. Vacuolated, you see vaculations here. Vaculations in the glomerular basement membrane has GBM has occurred, right? So you can see a lot of vaculations. This is just because of injury. Deposits found in the GBM, right? Increased synthesis of GBM protein components, and there is formation of GBM matrix. So deposits in the endothelium may be immune complexes, most common fibrin, amyloid, glycoproteins, abnormal fibrillary proteins. These deposits may be found in the endothelium, epithelial, or within the lamina densa of the GBM. So if there is immune deposit in the glomerular basement membrane, so it will be either in the lamina densa or the outer layer, or lamina rara externa or lamina rara interna. Lamina rara interna is also called subendothelial, right? Subendothelial deposits, or lamina rara externa is also called as subepithelial deposits or epithelial side deposits in the epithelial side. Subendothelial, subepithelial, and uh, lamina densa GBM. I think you got my point. So increased synthesis of protein components of glomerular basement membrane, as in diabetic glomerulonephritis, that will cause crescent formations, right? So here it is crescent formations, lamina densa interna, lamina densa externa. Interna is subendothelial layer, externa is subepithelial layer, and there is proliferation of the parietal epithelial layer in case of parietal epithelial layer in case of diabetes mellitus, that causes crescent formations. These are called diabetic crescents, as in diabetic glomerulonephritis. Lot of protein is synthesized. They deposit in the glomerular basement membrane and elsewhere, and they cause a lot of problem in the filtration of the blood that comes in. Formation of additional layers of basement membrane matrix. There has been mostly subendothelial location. It ranges from poorly organized matrix to fully duplicated lamina densa. It occurs in the membranoproliferative glomerulonephritis. Membranoproliferative glomerulonephritis is a form of glomerulonephritis that is autoimmune, right? It is an autoimmune glomerulonephritis, of one of the autoimmune glomerulonephritis. So if there are deposits in this green area, right? if there are deposits in the green area, it can form, the GBM will get thickened and it will form new layer above the deposit and below the deposit. Are you getting my point? If there is an immune deposit in the GBM and it starts to proliferate, and if it starts to proliferate, it may proliferate above, or it does proliferate above and below the immune deposit. And that gives it a tram track appearance or a double basement membrane appearance. Although it is a single basement membrane, just it has, because of the injury, it has formed reactive layers above and below the immune deposits. So it has formed a two layer of two layers of this um, basement membrane, and that is what happens in the membranoproliferative glomerulonephritis. What is hyalinosis? We talked about protein that gets deposited, accumulation of homogeneous and eosinophilic material by a light microscope. Hyaline is an extracellular amorphous material composed of plasma proteins that have insulated from the circulation. They deposit and obliterate capillary lumen when extensive, right? So they are eosinophilic material that is amorphous, and it comes out of the plasma protein and it obliterates the capillary lumen, right? And if it is extensive, it will obliterate the capillary lumen. Look at the hyaline material. Look at the hyaline material. Hyalinosis is usually consequence of the injury to endothelial cells, capillary wall, typically at the end result of various forms of glomerular damage. See, so you can say when immunological injury is extensive or a diabetic injury is extensive, it will start to throw hyaline material that it will accumulate to an extent as to end the kidney function, nephron function, which is the end of the kidney function as well. So it depends on how many nephrons are functioning and how many have stopped functioning because of such injuries like hyalinosis. And if that is the case, most of or most all of them have got such a picture, then the kidney will not function at all. Endothelial capillary walls, or typically as a result of various forms of glomerular damage is there, right? Sclerosis is characterized by deposition of extracellular collagen matrix in mesangial area. It involves the capillary loops or both. So sclerosis, I told you, it is actually hardening. So if there is a lot of extracellular collagen matrix in the mesangial or in the capillary loops or in both the places, it will result in obliteration or some or all of the capillaries, and this will cause no function. And can it be any useful function? Can there be any useful function here? No, I don't think so. If there is a crescent formation, there is so much of hyaline material deposit there, and it is hardening of this area as well, then no, there will be no function. Let us go through the entire substance once again, and let us see if we understand properly, and then we will go on to the diseases. So today we have done the glomerulus, and we are finished with what a glomerulus looks like, how things get injured in the Bowman's capsule. So we said glomerulus is, and the kidney nephron is basic functioning unit, and we said glomerulus is the basic filtration area, right? And for the filtration, there is some special features of the endothelium of the capillaries in the There's some special features of the capillaries in this glomerulus. Khas features can just let us summarize the whole thing. So, what are the special features? There are pores in the endothelium. There are two capillaries that the glomerulus ends up in two capillaries. It is surrounded by two capillaries going in and going out of two capillaries. It is a high pressure area. So any change in the pressure will strongly affect or damage the glomerulus. If there is change in the generalized body blood pressure or just the pressure of this area because of some hyalinosis, sclerosis, or whatever inflammation is there, that will massively damage the glomerulus. And then we have also gone through what damages can occur. Just looking at this diagram, we can see there are mesangial cells that have special features. These are phagocytic cells, and these have got features of muscles as well, smooth muscle cells. So these special cells, when they go undergo proliferation, it means there is some kind of inflammation there. Other than that, their cellularity can be increased because there is accompanying inflammatory cells. Inflammatory cells can be monocytes, neutrophils, lymphocytes, right? So the usual inflammatory cells. And then we said that there is some degree of proliferation in the podocytes as well, and the podocytes through foot processes that are called pedicles. In between the two podocytes, there is a membrane that forms a diaphragm, right? And this diaphragm is formed by the nephrin. Nephrin has got an extracellular portion which forms the diaphragm, and a small intracellular portion that is connected to the molecules inside the cells. Three molecules inside the cells, right? Protein associated molecule, CD molecule, and the third was actin molecule. So that is how that is how the filtration barrier is formed by the slit processes here in the endothelium fenestra, 70 to 100 nanometers. Then there are negative charges here. Second barrier, third barrier is the podocytes, and the slits they form. Slits are 20 to 30 nanometers in diameter, and then there is a diaphragm between the two podocytes, and that also forms a barrier. Once those barriers are broken, there is a lot of protein deposits inside the whole area. These protein deposits will call on more inflammatory cells, and the cytokines will cause damage to the parietal epithelial layer. And what parietal epithelial layer does in reaction is it causes proliferation, obliterates the urinary space, and finally it will form a crescent, and it will narrow down the area through which the urine flows out, right? So this is what will happen. So this is the diagram showing in elaboration what mesangium looks like and what podocytes look like, and this is what endothelium looks like. This is a better diagram showing interdigitations. You can interdigitate both of your fingers and talk of the pedicles there that interdigitate your fingers, and in between your fingers is this blood vessel. Holding between your fingers is a blood vessel. Right. This is how a structure is formed. This is the nuclear area, right? Podocyte. This is the diagram in your book. I explained it to you. Pink area is the basement membrane. When it has got diseases, you will see immunological deposits. When you've got immunological diseases, you will see immunological deposits either subendothelially, subendothelial, or in the dense layer of the basement membrane. I think we are done with most of this. We have also revised, yes, the collagen type of the kidney. Collagen type in the GBM is type 4 collagen. So these are the other molecules present there, glycoproteins present there, and actin, fibronectin, polyanionic proteoglycans, mostly heparin sulfates. They are all negatively charged, right? So we are mostly done with this, and I hope things are quite clear. If you go through your book, you might still find some difficult portions, or if you do find some difficult portions, please do contact me so I can explain and clarify all of them. And in case you do not read it, I will still please advise you to read it, because in this way, when you are sitting alone reading a lot later, you might find it rather lost in the book. Right. I think now that we will end lecture here. Thank you very much. Thank you for your patience, and thank you for listening the lecture patiently.